Hi there. My name is Professor Matthew Schmidt, and welcome to Genetics. In this session, we're going to conclude our discussion of the eukaryotic chromosome by talking about the organization and types of sequence elements present in particular, eukaryotic chromosomes. Now, we could have called this section Genomics, and the reason I didn't is because it's really not a full exploration of genomics. But we should mention what the idea of genomics is and why this relates to it, and furthermore, why I put this part along into this section about the chromosomes. So, eukaryotic genomes, this field of genomics came about because it's been known for quite some time that the entirety of the DNA in a particular organism doesn't necessarily all have to do with coding for proteins, which is certainly what the gist of genetics was for a long time. And it's still very, very important. But with the completion of the Human Genome Project, it became very clear that there are all sorts of different DNA elements aside from genes in the genome. So genomics is the detailed study of the entirety of the whole DNA that's around. So a couple aspects of that we can take a sort of quick look at. We're certainly not going to go, you could probably take a whole course in genomics. But one thing is, it turns out when chromosomes are stained with appropriate dyes, and you may have seen pictures of this, various banding patterns are observed. And if you're an expert, you can sort of tell what those banding patterns mean. But one general interpretation that we can say, a lot of times when you see these pictures, they'll be of metaphase chromosomes. But even during interphase, some areas are more darkly stained than others. So the dark staining gives rise to the idea that there are two basic categories or types of chromatin, and they are euchromatin and heterochromatin. Euchromatin tends to be not so densely stained. And the density has to do with how much DNA is in that particular region. So the interpretation of the fact that it's not densely stained is that these are single copy genes, and that tends to mean that these are where the protein coding genes are. They're not very compacted, which is how this goes along with our previous discussion, because by and large these genes have to be sort of open because they may be interacting with the machinery of the cell. So euchromatin is not densely stained. If you had to make a real simple one-to-one correlation, we would say that protein coding genes are found in euchromatin or vice versa. Whereas heterochromatin, on the other hand, is very densely stained, which implies that it's compacted, and that also implies that it's an inactive region. Now, that doesn't necessarily mean it does nothing functionally. It's inactive in the sense of it's not getting transcribed, replication's not occurring at that particular point. So usually, and we're going to spend a good amount of time on this, they contain repetitive sequences and not so many genes. So to make a connection of something we've already discussed, centromeres and telomeres are both found in heterochromatic regions. We know that centromeres and telomeres have functions, but generally speaking, they're certainly not something that's getting actively transcribed, for example, all the time. They have a different type of a function. So euchromatin versus heterochromatin. Now, this is about eukaryotic chromosomes, but it's instructive to think about how bacteria are in this respect and then compare that to eukaryotes. So in prokaryote, the genome is very streamlined. It is made mostly of genes. There aren't a lot of extraneous sequences there, and sequences tend to occur only once in the genome. So for prokaryotes, the whole genome, even though the study of genomics is relevant, largely there are only single copy sequences. If you went into this in great detail, we might have to modify that, but that's the basic idea. There's not a lot of what you would call heterochromatin in prokaryotes. You don't even really call what's there euchromatin because it's the only thing that's there. In eukaryotes, we have a whole different situation going on. So there are definitely single copy sequences, but as much as half of the entire genome is composed of repetitive sequences, which can be present in a seemingly ridiculous large amount of copies, as many as 1 million copies. I'm going to try not to say this over and over again, but there's great variability depending on the eukaryotic organism in terms of exactly how much of the genome is made up of this, how many copies are present, etc. So we're going to use humans sort of as our organism of choice because we are humans. If you're not, send me an email, okay? Anyway, the field of genomics does study the genome at large in great detail, so we're going to study just a couple of aspects of this that relate to our eukaryotic chromosome idea. So remember, we've got euchromatin and heterochromatin. I'm not saying they go together exactly, but there is an idea that euchromatin has single copy sequences, while heterochromatin probably has repetitive sequences in it. Now, we want to get a little bit of experimental information in here. So how did they even discover repetitive sequences? And we can't go into great detail about this because it would take quite a long time. But there's a process, I think you'll get the idea, that involves denaturation and renaturation of DNA. And the kinetics of that, which is just a fancy way of saying the time frame of how long it takes for denaturation and, in particular, renaturation to take place. So these type of studies really were the way that not only repetitive sequences were discovered, but that different types of repetitive sequences were initially characterized. And also, as you see here, demonstrating their relative abundance in different organisms. So it's a pretty, I wouldn't say simple idea, but you guys are becoming genetic experts now. So in the process, say you take human DNA, you could do it with any organism. You cut the DNA into pieces using restriction enzymes. So you have basically thousands and thousands of pieces of the genome hanging around. You heat it up so that the strands denature. So you have single-stranded pieces of DNA around. And if you cool it down, they start to reanneal is the word, or renature, meaning they find their complementary partner sequences and go back into the more stable double helical conformation. So reannealing, renaturation, the basic idea is we're going to heat this up and let it come back together. But the reason we cut it into a bunch of little pieces is if you just let the whole genome do this, it would not be as instructive for reasons you're about to see. So I hope this makes sense. In general, single copy sequences will renature at a predictable, relatively slow rate. When I say relatively slow, it's because what is fast and what is slow? It's relatively slow compared to some other elements in a eukaryotic genome. But the reason is because, say there's one gene here, if you denature that, first of all, since eukaryotes are in fact diploid, there are in fact 2 copies of each gene, right? But they may not be identical. There are different alleles. But put that little variable out of the picture for a minute. We're thinking that here's a gene, and when it's going to find its partner, its complementary partner, out of all these, who knows how many, millions perhaps of pieces of DNA that are floating around, only one of them is its partner. So the kinetics of this is they sort of all just bump around randomly until they find their partner, and then they renature. So since there's only one correct partner sequence for single copy sequences, it's a slower process as they're trying to look for their partner. So it was found out that certain sequences renature much more quickly than you would predict based on the single copy kinetics that you could, I'll show you a picture in a minute. And it was realized that these sequences represent repetitive DNA. The higher the copy number, the faster it will renature. Because think about a repetitive sequence as opposed to a single copy sequence. When the single copy is looking for its partner, maybe 1 out of 1 million will be able to pair with it. But if you're highly repetitive, I'm just making up the numbers, but this sequence, there might be 1,000 or 10,000 identical sequences around. So you're going to be finding your partner much, much more quickly, right? So it makes sense, and if it doesn't, think for a minute or go back and